Hello, everybody. My name is Jordan Clemsen, and today I'm going to be presenting my review of this paper titled "Environmental Microbes Drive the Chemotactile Sensation in Octopus." Uh, I think it should be octopuses, but either way. Um, Before I get into the methods and results of the paper, I'm going to go over some relevant background information that applies to the materials presented in this paper. So just really brief review of the microbiome. They list bacteria and archaea, two differences between the differences between the two, aside from the peptoglycan that is present in the cell wall of bacteria, but not present in the cell wall of archaea, pseudoglycan itself, as well as the cell membrane of ester bonds in bacteria and ether bonds in archaea. So two distinct differences that make up the animal kingdom. So when they're referring to those different subtypes of the microbiome itself, it'll be it'll be keep in mind different kingdom as opposed to bacteria, archaea, and animalia, if that helps. Um, they Another strong component of microbiome that they don't really address, but one to keep in mind is bacteriophages too also do regulate the abundancy of these bacteria involved. There's also fungi that also are defined loosely from the chitin in their cell wall, which adds to the structure. They typically produce chemicals that are poisons to the bacteria, so they're a competing factor in both space and nutrients, and their fermentation has a adverse effect on that count. So it's something to keep in mind as well. Um, I mean, just random thought, a little bit of a tangent, but penicillin derived from mold. So that's an example. Protists also present in the microbiome itself. They're going to be much larger, but they will impact the microbiome itself. So something to think about too in the background of this paper. The images I have here
I think Paul
are,
has a question.
oh yeah, sorry.
So can you tell what is
Yep.
protist?
Protist is it's basically, if you break it down, so it's how I remembered it when, because it's basically the other category kind of in the animal kingdom. So protist itself is it's before animals, if that makes sense. So typically this is probably an extremely diverse subsets, like an amoeba is a protist, hydra is a protist, but they're more complex, if that makes sense. Some of them have cells inside of them, but they're still like, tiny. Like maybe I could show you a Volvox afterwards, but they have like they're like, oh, pardon?
Are they like advanced bacteria?
No, multicellular, right?
Yeah, they can be.
Some
They can be
things.
multicellular. They have different there's it's kind of the other category really. That's why it's kind of interesting. There's things like Volvoxes, there's things like amoebas, those fall into that category because they kind of have more
Oh, but
complex.
maybe in one cell.
Yeah, again, it's other. Some do, like Volvoxes are multicellular, if that makes sense. So again, I always think of it as the other category that doesn't really fit into the rest of this. Good question, because I it's kind of a hang up on mine. That's why I think it's the most probably my favorite part of microbiology was that because you see some weird stuff that is kind of terrifying, but they're also the largest, so you can actually see them. That's one thing I feel like is generalized. So you can see them underneath just a light microscope. I remember as a kid, I could see them in the water when I'd hold them up. So it's like I can see with my eyes if I like squint. But yeah, malaria, for example, is caused by a protist, if that helps. I hope my lack of a
It gives an idea.
definition. Yeah, there's a lot of rules, but I think it's they're pre-animals, I would say, is what I would say about that. Um, Again, distincting between pathogenic and non-pathogenic, I think, is the proper terminology that is the nomenclature it's currently used to, for example, I mean, I'll go over this when I show the results, but they have bacteriocytes, which are uh, typically non-pathogenic in human beings, but they're also apparently present in octopuses. I'm going to point that out, as opposed to pathogenic microbes itself. Um, and also I want to point out too that the relationship to the host, I guess, from the nomenclature of pathogenic, non-pathogenic, could be extremely vague. For example, hemolytes, good gosh, which are worms that we digest in our developmental stages of our, uh, I learned this last semester too, it was fascinating in neuroimmunology, that we ingest as is a developmental stage that you typically in 3 months of age that we eat dirt and if that makes sense and they're parasites that I mean, they're typically parasites, but they are kind of they're allowed in the system. They're in all of us, but they're allowed in the system because the immune system can train itself to recognize that this is a threat, if that makes sense. So they can use it as an example. So there's been
Cannot train itself.
no, so it could train itself. So like 3 months of age, subjects who are not we learned this last semester in neuroimmunology that are not present to hemolytes have a significantly higher risk of autoimmune disorders. And partly they did find a similar mechanism. I can't remember the mechanism, but it was like it had something to do with the cell wall and how they can identify the antigen and then specifically apply that and generalize that in a more generalizable contexts. Otherwise, you have a higher chance of autoimmune disorders in the brain, like multiple sclerosis. That makes sense because then you're attacking the Schwann cells. But anyway, so I'm just going over that very briefly. I can probably talk about that way too much, but the relationship to the host can be very vague. For example, hemolytes, I guess, technically a parasite, but we need them because otherwise we have autoimmune reactions. I use that as an example. Um, or there's like really beneficial stuff like bacterotites, which help us or lactobacillus, which helps us digest milk, which is pretty important because we need maltose, which is the sugar from the milk. Um, Going on, and then just really go over the background. There's two more sides of the background. I'm going to see if I can go through this really quick. So they discuss a lot about breeding behavior in octopuses. So the females typically die after the eggs hatch. These organisms are use the R strategy in terms of reproduction, which means that they have a whole bunch of offspring, but and most of them will probably die, but they have it's like a shotgun approach, if that makes sense. So as opposed to a K strategy, which is just animals have like one offspring or like fewer, if that makes sense, but then they invest more time nurturing them. Human beings are probably the best example of K because we are so helpless as infants as opposed to octopuses. However, one thing I want to point out that should be noted is octopuses do put a significant amount of nurturing into their mini offspring to the fact that so they actually die. They actually stop eating, so much so that they starve themselves and have a death spiral. It's called where their hormones fluctuate once the eggs hatch, and they just kind of die. If that makes
What's
sense.
the evolutionary
Purpose, because so what happens when the octopuses give the eggs is they stop eating and they stop the predatory behavior, so they can just basically hold the eggs, blow on the eggs nonstop, so it gets rid of parasites, if that makes sense. So it's basically kind of a caveat. That's why brought it up. There technically are strategy organisms, but the octopus puts a significant amount of effort to the point where it dies. So I'm not sure what to say about that, but uh, that's why I brought that up. So the evolutionary purpose is um, so the octopuses don't have parasites, if that makes sense. And so the eggs can not be eaten because they do kind of join the phytoplankton, if that makes sense. And lots of predators like that, and obviously, so it's feeding. Nine brains, which we'll go over too, and they mentioned that briefly from a neural net. The brains and the tentacles themselves for each tentacle is referred to as a ganglia because it's technically part of the peripheral nervous system. Um, it's also interesting that these neurons are all of them are extremely large in diameter, so none of them are myelinated, which is interesting too. They have supposedly evolutionary life cycle, which would go into the behavior analysis of the paper itself, and they're also opportunistic hunters. Um, as present, except for when they're, I guess, breeding, because then they basically devote all their uh, energy. They don't even sleep into eggs, so they kind of just do nothing but that and die. Um, I put this in the image on the right. This was back in 2021. I think earlier, I think it was 2008. There was evidence that a giant Pacific octopus, which is what's listed here, uh, ate a shark in an aquarium, and that was previously not thought of. So they're typically opportunistic hunters that will eat things if they can, if that makes sense. And then what's really of use to this paper is they have chemotactile suckers. So we as humans, and this is also why it kind of is important for biomedical science in general. We know very little about the sense of gestation. We went over this extensively in our neuroscience class last semester, which I'll review. But so the mechanisms are similar, if that makes sense. And the whole point of why this paper kind of looked into using octopus as a model is due to the chemotactile suckers that essentially just which act as taste, but are also touch based, if that makes sense too. So it does interact with the environment quite a bit, and then will also interact with the microbiome and such. Um, it is also important to note that the bacteria underwater tend to form little like clusters and slimes due to the liquid that they're encased in, if that makes sense too. So they're more densely packed as opposed to in the air where it's more free floating. So and then just to briefly review gestation, because that's like essentially what they're looking at in this paper is the microbiome and how it interacts with this sense of gestation. So in human beings, we have five tastes. Does anybody know what the five tastes are? They want to shout them out at all? It'd be fun to interact.
Sweet, sour, bitter.
Umami.
Yeah,
Okay, what else?
Umami, like savory.
salty.
Salty.
Yeah. I was like I always forget that one too. We actually don't know. So sour we know it's an ion it's an ion channel. Bitter we it's funny. Salt we don't really know the receptor. I know there's a lot we don't know. This is like the apparently I had the same reaction to Sergey last semester. I was like there's a lot we don't know about this, which I guess is why
I mean,
it's
it's so basic.
Yeah.
Sodium channels and all of that.
you think it would be a sodium channel. I was I don't know, did you have the same reaction I did last semester? I was like, oh wow, you know nothing about this. Great. So sour, unami is just protein sensing, so that's probably evolutionary purpose if that makes sense. Sour we know it's ion channel. Salt we don't know the receptor, and bitter we don't know the receptor, but we know it's something with poison if that makes sense. So yeah, so think about that with coffee, I guess. So five tastes there. Spicy is not.
Slow death is fun.
Yeah, yeah. I mean, if you can't,
Coffee.
if you like coffee, it's a slow death. But poison scents usually adapted from environment. The spice itself is
That's
not
why kids don't like coffee. They're so about like
really that. Maybe that's why. Yeah, I don't know. That's a good. I don't know.
and beer also. It's like it's bitter taste.
That makes
kids
sense.
don't like, but then when you start auto pop or auto whatever, when you start killing yourself,
Yeah.
sort of the second
Apoptosis,
half
and we just
yeah,
kind of
apoptosis, we kind of like start liking the
maybe we should do
the stuff that
Yeah.
kills you.
Wonder if there's been a neuroimaging study on bitter
So-
taste. Anyway, no worries. It's. I was just kind of shocked that we know like basically nothing about this, and it's pretty common. So spice is actually not a taste. It's just a combination of touch and smell that we have in our back in our pharynx. That's then processed mostly cognitively, that was interesting to think about. So it's a combination of those two, which
Also, it can be trained then.
yeah. Well, yeah, I guess the theory, yeah, because it's prefrontal cortex originally. However, what I'm gonna, but also, which I want to point out too, it originally it does, but it bypasses the thalamus though, so it's probably going to be autonomic. Smell is the one sense that will
You said
bypass.
cognitively, and that's triggered. Oh,
Yeah.
it's like you.
Eventually, it is downstream. I should say. I should say way downstream, if that
Okay.
makes sense. It's because we have the stimulus, but it's like afterwards, if that makes sense. So we have the reaction, and then we think, oh well, that you taste, but we had the reaction before we were cognizant of it. Do the pathway. Every other sense will go through the thalamus. I'll discuss the pathway really briefly on the taste too. But yeah, feel free to chime in there. We had this last semester, it was fun, but and then touch is just free nerve endings, if that makes sense. It's like a pain sense too on top of it. So combination of those. Taste itself as we have human beings have papilla, octopus something similar, where it's like well, structurally similar. I don't. I mean, part of me just wants to call it the same thing, but it's basically a crevice where underneath the crevice itself there is the sensory. There is basically like this thing called the taste bud, which is like a spear that has all these different cells in there, and that's where all these interactions occur. These also regenerate like every 6 months, I think it was through, right? Like last semester, 6 months, taste buds, 6 months. I remember the mouth had the most regenerative nerves, if that makes sense.
I said smell. Okay, interesting.
Smell was like second. It was that one was like year. I thought the same thing, but then the taste was more. And part of me wonders, and I asked this in class, part of me wonders if it's because of the microbiome, because like a lot of the mouth microbiome itself is really pathogenic. That's why we want to Like that's, mouths are bad. They're pretty gross places. So and our body works like tirelessly to try to combat that, whether it's mucus, whether it's inflammation, whether it's tons. So there's a lot going on in the mouth. But anyway, so that's the taste bud. Typically, the pathway that we discussed last semester is the gustatory G protein. Then once the taste bud is activated, depending on the receptor that we don't know really much about, imaginary receptor is activated. The G protein's activated. It goes to through cranial nerves 7, 9, and 10, and then that travels to the medulla, not the part of the brain, not my cat. And then it would well before the medulla it goes to the nucleus accumbens, and that's the emotional reaction. Then which is also activated in alcoholism. Fun facts. That's where food addiction can kind of come in. And then it goes to the medulla, which then goes to ventral part of the thalamus, and then eventually all the way up to the gustatory cortex, and that's where we kind of are cognizant of the food, if that makes sense. So, yeah, sorry, I can move that too. Sorry. So yeah, that's just a relative faster in humans. In octopuses, it's kind of similar, where they have a suction cup, which is kind of the crevice of the taste bud. Once that part of the suction cup is activated, it fires a similar ion channel that goes up to the base of the arm, and then it's processed there, and then processed in the brain, if that helps. So I want to go over that. Now that I went over that, because it's why it's a relevant animal model, I'm going to go over the base of this paper. So before I go into what they did, I want to instruct the hypothesis. Essentially, what they're attempting to do with this study is they were trying to explore microbiomes and sensory experience, but they in other animals, if that makes sense. And they use the octopus because they interact a ton with everything using their suction cups basically to taste their environment. And so that sensory experience just kind of interacts a lot. You think you would touch a lot of different things that are kind of gross. And so microbiome would be interesting there. That's why they used a relevant model for that. Now that I'm going to this, here's how they explore the paper. This is from the paper itself. So the California two-spotted octopus, also known as octopus bimacules, Ides, I think. I'm not pronouncing the last part right, but it's essentially caught. Two-spotted octopuses were caught in La Jolla, California, basically housed. They followed the Harvard, what is it, animal model ethics, if that makes sense. They also which is essentially just that both sexes, male and female, they have 12 hours of light and dark cycle, fed regularly, and given adequate treatment. So I mean that wasn't a factor including this. They were their experimental model or their experimental design was essentially that the crabs they were giving basically crabs and decided if they both decayed and living crabs. And then they wanted to measure their reaction based on multiple and then they have respective controls for each one of these, which I should bring up as well. But I mean, we don't have time to go over every single one. But they had crabs living and deceased, and then they looked at how the octopus interacted with this crab, whether it's decaying or alive, or the eggs, whether these were actually mothered eggs or whether they're just false eggs, if that makes sense. And then they monitor the behavior and a whole bunch of other stuff, which I'm going to get into. They measured a lot of the behavior, I should say, a portion of this study through video scoring egg rejection. I don't really know what that system is. I'm assuming it's similar to a Likert scale. It sounded like it was ordinal. If that makes sense. They used predation decision, which is, I guess, something that they use from a camera, which is interesting too. So it's more subjective, I would say, maybe a categorical variable. And then they also measured floor expiration, which was the used interval data. Well, I guess it wouldn't be interval because there's not going to be a zero. Well, I guess it could be because maybe they didn't. So interval data because they described it as duration and whether they touched the ground, if that makes sense. And that's measured from video camera. So that was utilized. How am I doing time-wise? Am I doing okay?
Yeah.
Okay.
34 left.
Oh, okay, good. Okay. And so that was used. The images on the right are from the study. They just one of the tiny figures that they use. It's a picture of the two-spotted octopus in California. In La Jolla, they're pretty rare. I think I saw a cave one when I was there in November, but yeah. And they also fed them fiddler crabs, I should say. That's the type of crab that they used, pictured. What they found, so what they did to measure the microbiome is they used osmium treated, they put osmium on the crab cells themselves. They said they used SAM, which was interesting, and prepared them to dehydrate through ethanol and to not kill the bacteria themselves. But they used one of the main techniques they use multiple. I'll go over them in a bit, but they use the scanning electron microscope. It's as long as I mean I'll go into the oh my gosh the mass spec that they used as well as the cultured strains. But the scanning electron microscope they extracted the bacteria, which I'll go into a bit too. But they found that they're more sorry, and they applied more eggs and crabs with the rhizin itself and swabbed the 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 residue after the octopus touched it, before and after, for and used a 16S sequencing and the culture, but I'll go into why they cultured in the next slide. They used, I think it was I forget what they used for clustering taxonomy. I don't, it wasn't kaiju, but the reads were denoised using the data too with Q, I can't say that right, but QIIME. And then they also signed clustering as well. Oh yeah, it was BLAST. These,
So essentially, they're kind of looking at how these octopuses interact with the crabs.
Yeah.
With different microbiomes.
they're trying really hard to control for a lot of different variables. So they looked at behavior, they looked at, and then they also so they looked at behavior. Yeah, they tried to see how they interact with the crabs, and then with eggs, because they wanted to also measure brooding behavior, if that makes sense too.
So are crabs a predator too, octopus?
No.
Or prey?
Well, I mean, if they're eggs, that they're going to be a predator because they want to eat that. But octopuses they typically love crabs. So that's what they usually use for positive reinforcements in a lot of these studies. So they used it as food, if that makes sense. But they also had like decaying crabs, and I don't want to go too much into that. I mean, I kind of can on this because you can see in this figure that they had a lot more cyanobacteria, which is typically what's also in humans not good. It's pretty toxic. Like we don't really like cyanobacteria. It's great for pond water, um, in moderation, but not great for human beings. Um, and so, well, in excess, but it's really high. It's low tolerance for that. They found them a lot more in once I go over the methods of how they did. If you can see from the figure here, they found a lot more cyanobacteria in the rejected crabs, in the decaying crabs. Uh, and on the octopus, if you can tell, the ones that did not eat the crabs, you can tell that there was, and you can tell, I don't think this is the figure that has yeah, no, it is. It's the one that has the body parts on it as well, too. So you can tell where they swabbed, and what culture they found based on the assigned taxonomy. I'll point out certain ones, sorry. Protobacteria is non-pathogenic. Bacteria I mean, that's the one I talked about. That's extensively found in our human being gut. That one is pretty nice because it functions as a placeholder for stuff that could be worse and also helps with digestion. Um, what else we could kind of see on there? That's also good. Cyanobacteria is pathogenic usually. Um, and so, I mean, if that's an example, they found multitudes with some of different bacteria that look very different. As you can see on the heat map, well, not heat map, but look different based on where they were found on the octopus. I'm sorry. I hope that answers your question. So, basically, crabs, and then eggs, and then decayed crabs and living crabs, fake eggs, real eggs. So that makes sense.
Okay.
So it's like a tree. So hope that helps. And then they did a whole bunch to each one of these to basically weed out any confounding variables. So they used, because they also looked at behavior, but the behavior itself was just as I just went over, those things were used to just the videotapes and the durations were used to assess behavior. I'm not as interested in the behavior, but I think it's important because it's how we interact with the microbiome and it's how these microbes are shared. Sorry, so then they ran a Promova and Perm Nova, and then breakers on the taxon results afterwards. One of the main things that they wanted to look at too was the CR1 ligand, which is an ion channel in the octopuses, and they were kind of curious about how that interacted because it's hypothesized to be used in gestation. and they wanted to essentially look at the ligons and see if they are similar. So so to assess what ligon was actually attaching to the receptor, they culture the bacteria from the previous slide. So they basically rise in the bacteria and then kind of grew them back out, if that makes sense. So they could study the bacteria itself after the after the octopus has interacted with the crabs and the eggs. Um, from that resin that I discussed, and then they elute the products to see what the bacteria were fermenting, if that makes sense. And then separated and retested just to double check to control to make sure that they didn't have a sampling bias in the way they eluted for the bacteria. They also identified the active with another control. They wanted to identify the active compounds using the high res mass spec as well as the NMR, if that makes sense too. So they wanted to look at different models. So they looked at basically a lot of controls to make sure that they're actually getting what they're getting. They also looked at metabolites, but they used UV for this to identify each one of the metabolites. If you can see on the right here, these are the figures that they presented with the metabolites that they showed. It's just pretty straightforward. It's pretty it's what you expect from the bacteria that they showed. It's a pretty um cohesive story, if that makes sense. Uh, they also then to uh to look into the arms itself, they wanted to double check if the octopuses were healthy, if that makes sense too, and see if that actually played any factor on the interaction of chemotactile taste and the microbiome. So to measure that, they use electrode recordings to measure spiking, which is just pretty straightforward, where they can look at ooh, sorry, um, neuronal mechanisms and if there's an action potential fired when the octopus is doing certain things in the video. They used RMS for this to measure the spike timing and define it. They also randomized too to make sure that there wasn't an extra step in the process. They also use video recordings at I think it was 5 frames per second. That can't be right though. And use stack-based approaches to measure this. This has been something that they've been doing a lot in the, I don't know, octopus cephalopod research field. I think it was a separate paper where they used deep learning on octopus tentacles to decode language, which was interesting. So there's been like some merit to it, but I was skeptical about using the arm movements as a behavioral uh, um, indicator. And they also measured sucker adhesion too to see if that was actually a deciding factor to see how long based on duration when they touch the seafloor, if that makes sense. And part of the way that they did this is they had an auger that they used. So when the octopus that they basically had the tentacles, and they obviously after they're euthanized had the tentacles placed in the auger, and then they call it lift off, but it's like they lifted up the tentacles and saw if the suction cups ripped off, if that makes sense. And that's what they use to measure count for adhesion, if that helps. Um, and they reported the results, if you can see too. But again, it's positive results, and that also kind of backs up what they're saying. Um, and then they disguise that in their discussion, they basically say that discrete compounds for the microbiome are indeed interacting to detect the environment. They say specifically in verbatim, here we discovered the microbial ligons encode diverse cues for both predation and brooding. So seeing, positive results, which is pretty straightforward. And they also said that the CR agonists that they're looking at with the ion receptors are enriched. And they note that their conclusion is based on the one group's reaction to the decaying crabs. And I am also going to merge my critiques with this paper too. So they have a huge section that they went over the limitations of the study. I think that's the most interesting part of this paper because it does kind of suggest that there's a lot of future study that could be researched after this, but also maybe the quality of the research itself. They said that it was limited by the culturing method. They didn't specifically say that there was cross contamination, but they brought it up multiple times, which makes me think that maybe there was an intern that messed up somewhere along the line, and that probably is where they're getting. Yeah. What's interesting, they kept talking about cross contamination. I thought that's weird. Why are you talking about cross contamination when you which is probably why they had so many blocking mechanisms just in case. I would be curious to see if there was retroactive, controls if that makes sense, just to make sure it wasn't caused by something else. Um, which would make sense because they're finding a lot of human bacteria on the octopus as well. I'm curious to see if that was interaction. One critique I have too is this is only one location. It's La Jolla. They only got the octopuses from La Jolla, California. And if you've ever been to La Jolla, California, in the cove where the octopuses are found, it smells like oysters. So it's just riddled with bacteria. Um, I was just there in November, and I still did smell my nose, I swear. So I think that was kind of an issue, especially since there's other type of octopuses that interact differently with the seafloor. Mimic octopuses use different parts of their body to touch the seafloor too, which would be interesting. Coconut octopuses essentially basically live inside a an oyster shell, and like hermit crabs walk around with it. And then my favorite type of octopus, which I'm kind of more interested in, Octopus vulgaris, or the giant Pacific octopus, which I showed earlier, is usually the most intelligent, but also has the predation. And I'm curious to see if the microbes differ in that context, especially since they aid in fermentation, and it'd be interesting to see larger animals. Also, the frame rate is really limited in the camera, and I'm not entirely sure that they can see really quick reactions on 5 frames per second. And yeah, that was my thought too. I was just thinking like that's not gonna do much. What
That's
what
what cats think about it.
yeah, that's probably is like, why is he so slow? Um, my cat bites my leg because they don't walk fast enough. Um, but yeah, so I thought it was an interesting paper. I really liked how they handled the microbiome. I think that was the really thorough part because it's really I do think there's a larger issue with the a unit of measurement. Uh, so that's why I think people have to really be serious about what they're using to define a taxonomy. As I went over my previous slide, cross-contamination can happen, and there's very subtle differences between huge differences in species or even kingdoms, if it's archaea or bacteria. And I mean, just an Esterbon, if I go back to the slide here, I'll go back to it. This is why I went over this. It's I mean, there's just one. I mean, there's the R group is, if you can see an Esterbon and an Etherbon in a plasma membrane, there's just what a oxygen that's separate from the R group, and it's the orientation. And I feel, and that could be, messed up really easily. And that tiny mistake could mean the difference between a kingdom, whether it's an archaea or bacteria. So I mean, I think it's an issue. And
We are here.
we're eukaryotes.
Yeah, but I mean, we
Archaea
are
would be like
descendants of archaea, not from bacteria.
uh
It's an archaea ate another bacteria and we became eukaryotes.
that's been supposedly because archaea don't that's based on the theory because they're usually not pathogenic. However, I would completely argue because there's been some people who have like really gone in like weirdos who like really wanted to see if it was pathogenic and like tested on themselves and then got infected, and it's like, oh yeah, of course. So typically they're not, yeah, because like, so for those of you who don't know, archaea are extremophiles that usually find them like sulfur geysers out in Yellowstone, if that makes sense, because they live in these extreme temperatures. Like their body operates like extreme pH, extreme temperatures, if that makes sense. And so they don't really. My argument would be, well, our body is not that hot, so I don't think it would want to. I don't know why I would want to do that. The fact that the Yellowstone just exists and there's nothing there that can live without the heat creates like a vacuum, and that kind of is what the archaea wants. So they typically are a non-pathogenic, so they don't really, I mean, in my opinion, have to infect people because they have all this open space that they can just interact with. CRISPR technology, if anybody heard of CRISPR-Cas9, it's an archaea. The reason why it works so well is because the temperature differences. That's why, because their body their polymerase action functions differently at room temperature just because of the temperature difference that they are used to, if that makes sense. that's why it was kind of brilliant, because most of the archaea are these, I mean, they're extremophiles. But yes, so my point is that like a tiny difference like that can really mess it up, because while they're both prokaryotes and we're eukaryotes, um, bacteria and archaea are different kingdoms. Like it's archaea, bacteria, and animalia. So they're not. I mean, it's like to put that in perspective, it's like saying an animal is a bacteria, and it's that mistake. So I think the difference is a lot different. But anyway. So that's my discussion. Does anybody have any thoughts about this paper? I'm sorry, and those are my critiques. I think it's interesting from a microbiome standpoint and from an analogous evolution standpoint, because we can kind of see how the octopus's taste developed and evolved. Especially with, I didn't show that figure, but there was like an evolutionary uh, graph too that they showed, and I thought that was kind of interesting, because if the mechanisms are similar and we also interact, we evolved taste first too. Taste is a lot more useful in the ocean, because water goes through gills and it goes through your mouth, if that makes sense. I don't feel like it's as useful on land unless you're licking the ground. But yeah, so that was my thoughts about the paper. Anybody else have any thoughts?
Well, smell is more
Yeah,
useful in the air.
Yeah.
right, than- I mean, it's kind of smell is the same because it
Yeah.
kind of goes into the same bowl.
it's basically taste is basically smelling underwater. I feel like
Yeah.
so. Yeah, so I mean, I think it's interesting from that perspective of looking at how it developed, and I thought I was really taken back by how similar the our mouth microbiome help aids in taste, and how their microbiome aids in their own chemical detection. So, any
Yeah,
thoughts?
thank you, Jordan.
That was the end of it.
Any
Sorry.
other questions? Anyone wants to add something about the parrot? Yeah. I don't know. There are I have a lot of tangential thoughts because this is not like in the area. But I think
Sorry.
I may no, that's very interesting. I may hold my thoughts for offline discussion. Thanks, and anyone who watched this or listened to that. And maybe you have questions. I'm just wrapping up. If we don't have any questions, I think I will stop the recording and then it's just